I guess he's going to watch this tape at some point, so we should wave to him. It's a pleasure for me in your own stead, to introduce today's speaker, Stacey Rentschler. And Stacey probably doesn't even need an introduction here, because I think her outgoing personality and her sort of dynamic scientific manner have introduced her to probably everybody in this room, as well as the greater cardiovascular and developmental biology communities at Washington University over the past year and a half. So Stacey is the most distinguished graduate of Lehigh University in Bethlehem, Pennsylvania, which maybe many of you have never heard of, but it will soon be on the map. Take my word for it. She then went to Mount Sinai, where she was in the MD Ph.D. program, and she worked with Glenn Fishman, and it was at Mount Sinai as a Ph.D. student that she developed her interest in the cardiac conduction system. And I think that she has set as her career goal to become the world's expert in the programming and reprogramming of the conduction system. She, as a student in Glenn's lab, developed a way to identify cells in the conduction system and monitor their development. This is in the mouse heart, and then after she finished her MD PhD, she went and was a cardiology fellow at Penn, and then also worked in the laboratory of John Epstein. And it was there that she discovered that activation of Notch signaling in the myocardium results in two actually very interesting phenomena. One is an anatomical phenotype, the development of an accessory pathway that looks very much like the accessory pathways that you see in patients with Wolff-Parkinson-White syndrome. So it represents the first animal model of Wolff-Parkinson-White syndrome. And then in addition, she demonstrated that cells in the ventricle are actually reprogrammed with upregulation of Notch signaling to looking much more like Purkinje cells. And she is taking this work in her own laboratory to figure out the molecular mechanisms that regulate both programming in the conduction system and reprogramming of the conduction system. And it's a pleasure.
So, thank everybody for coming, and thank you, Jean. That was a really nice introduction. And can everyone hear me? I think. Okay. So, as a physician scientist, I'm very interested in how transcription factors and epigenetic modifications can influence the conduction system. And I've had a longstanding interest in the developmental pathways that regulate this complex network of cells and how they're induced to form during development, and how this system is patterned throughout development. And it's very interesting that we're beginning to understand that the same signaling pathways that regulate the formation of these cells during development can also influence adult diseases. So for example, the same signaling pathways that influence the PR interval and QRS interval are now being understood to modulate cardiac conduction and susceptibility to cardiac arrhythmias such as atrial fibrillation and ventricular fibrillation. So understanding how these components are formed and regulated, and the ion channel regulation by transcription factors, is important. So as you know, the cardiac impulse originates in the sinus node, travels through the atrial tissue, arrives at the AV node, where there's a delay in propagation before the impulse travels into the ventricle, and then it travels through the rapidly conducting His bundle, left and right bundle branches, and Purkinje fiber network. And this helps to coordinate the proper activation and contraction of the heart. So in addition to understanding the molecular basis of diseases, in addition, we can utilize these transcription factor networks in regenerative medicine approaches. For example, there are syndromes that originate from defective either development or reprogramming postnatally of conduction tissues. So for example, sick sinus syndrome in humans, which is characterized by sinus bradycardia or sinus arrest or sinoatrial block, or can have episodes of tachycardia and bradycardia syndrome. We really don't understand the molecular signals that regulate these disease mechanisms. So for example, shown here is an EKG from a patient, or a telemetry strip, where we see a P wave and QRS, and then we see an absence of P waves, which would be reflected by sinus arrest. And we treat these patients by implantation of a permanent pacemaker. And permanent pacemakers, or electronic pacemakers, are wonderful, but they do have some limitations. And some of the important limitations are the inability to augment due to the autonomic nervous system, for example, during exercise. In addition, in children, for example, as you get older, you need to have replacement of generators as well as leads. So there are some important limitations of permanent pacemakers. So if we could, I'll tell you a little bit about some of the future goals, hopefully, to be able to reprogram tissues into biologic pacemakers. So here's the outline for my talk. So first I'm going to tell you a little bit about cardiac development, because I think, though I know this isn't exactly the audience for that, I think understanding the basis for development allows us to understand the regulation of electrical programming in the heart. And I'll tell you a little bit about some of the methods that people use to regenerate cardiomyocytes, and specifically my interest would be in generating the conduction system myocytes within the heart. And then I'll tell you a little bit about the role of neuregulin and Notch signaling in conduction development. I actually won't tell you the story about ventricular pre-excitation just for the sake of time today, but I will tell you a little bit about some of the ongoing work that we're doing looking at reprogramming cardiomyocytes to conduction cells using Notch and Wnt signaling. So about 15 years ago, it was thought that all of the cardiomyocytes within the heart were initially specified in this first heart field. So this is a very early embryo, and the heart initially forms from lateral plate mesoderm here in this crescent in brown. And the heart will eventually coalesce into a linear tube, shown here, and then it'll form ventricular septum, atrial septum, and valves to form the fully mature heart. So we now understand that these first heart field precursors here in brown only form a portion of the cardiomyocytes that are eventually found within the mature heart. And there are, in fact, secondary heart fields, as shown here in green, which contribute to the venous and the arterial poles of the heart and are recruited at subsequent stages. And this is important because these different lineages of cardiac progenitors end up forming the different myocytes within the fully mature heart, as well as different subtypes within the heart. So the first heart field cells here in brown will eventually form components of the atria and the ventricular myocardium, whereas the second heart field cells here in green will eventually primarily comprise the right ventricular myocytes as well as the atrial myocytes. And in addition, we have epicardial progenitor populations here in yellow, which will contribute fibroblasts and smooth muscle cells to the heart, as well as cardiac neural crest cells here in blue, which primarily remodel the outflow tract and the large vessels of the heart. So an understanding of the processes that regulate the first and second heart field progenitors to form these different myocytes within the heart can help us to understand the electrical programming of the heart. So for example, as I mentioned before, the first heart field progenitor cells give rise to left ventricular myocytes, atrial myocytes, AV nodal myocytes, and His-bundle myocytes. However, the cells of the sinus node, as well as some of the atrial cells and the right ventricular cells, derive from a completely different progenitor, the second heart field progenitor within the heart. So if we want to regenerate this type of tissue, we need to understand these signaling pathways that make the first heart field cells become these types of cells, or the second heart field cells become sinus node cells. And so people have, others in the field, including ourselves, have tried different approaches to regenerate cardiomyocytes. And I'll just go through some of the different ways that you may regenerate cardiomyocytes in this figure. So first, one may take fibroblasts or a somatic cell from a patient and then reprogram them back to pluripotency. And then we can take these pluripotent cells and differentiate them into cardiomyocytes, which can then either be directly transplanted back into the individual or can be seeded onto scaffolds or hydrogels in tissue engineering approaches and then transplanted back. And this would require an understanding of how the induced pluripotent stem cells can differentiate into cardiomyocytes. And one of the limitations of these kind of approaches is that it's very difficult to generate fully mature cardiomyocytes using this approach. And for those of you who were at Michael Flamm's talk in the Cardiovascular Research Center recently, he discussed some of the ways that people are using to mature cardiomyocytes. But this process takes at least 3 months at current time. And it's a really good model for studying how these cells develop but it's difficult to make enough myocytes to actually transplant into an individual. So in addition, we maybe can take fibroblasts and directly differentiate them into myocytes. And I'll talk a little bit about this type of approach as well. But the approach that I'm most excited about is to directly reprogram the cardiomyocytes that are already in the heart, or some ways you can transdifferentiate fibroblasts into cardiac myocytes within the heart. So you don't need to actually inject cells into the heart or culture them external to an individual, but instead just tweak the cells that are already there to become the type of cell that you want. So first of all, I just wanted to go through some of the methods for lineage decisions during embryonic stem cell differentiation. So an understanding of development, it turns out that the same factors that regulate development, for example, in a mouse or human heart, are the same factors that we can use when we differentiate embryonic stem cells into cardiomyocytes. And so, for example, we need to not only understand the factors, but we have to give them at the right time, for example, to make a mesodermal type progenitor cell to become a cardiac progenitor, or a cardiac progenitor to become a primitive myocyte or nodal myocyte. And here's one example of the importance not only of the signaling pathway, but also the timing at which you activate it. So shown here are HCN4 positive heart progenitor cells within the heart, and we're labeling these cells blue using a genetic, and actually this was done by Sylvia Evans' group. They labeled these cells blue using a lineage tracer. And they did this at embryonic day 7.5, which is a very early time point in the mouse. And then they looked at embryonic day 16.5 to see where these HCN4 progenitor populations gave rise to. And they found that when you label these cells at 7.5, they become ventricular cardiomyocytes of the left ventricle as well as Purkinje fibers of the left ventricle. However, if you label the same population of HCN4 positive cells at embryonic day 9.5, and you look where these cells end up in the mature heart, they become components of the sinus node, the AV node, and the atrial septum. So if you take a progenitor population at different points in time, it gives rise to different parts of the cardiac conduction system. So we can utilize this understanding of development to perform direct reprogramming strategies. And the first instance of reprogramming that was published was by Benoit Bruneau's group, where they took early mouse embryos and used 3 cardiac transcription factors and reprogrammed these embryonic mesodermal cells into myocytes. So subsequently, tail-tip fibroblasts were reprogrammed using the same 3 transcription factors in vitro into induced cardiomyocytes. And other groups, there are several groups who've done this using different factors. And this process from fibroblast to myocyte is relatively inefficient in vitro. However, it's been shown that in an adult injured heart in a mouse, when using the same factors, we can reprogram these into induced cardiomyocytes at a higher efficiency. And what do we mean by efficiency? So the efficiency is, it depends on what you're measuring. So if you look at gene expression, you'll have a higher efficiency. If you're looking at functional changes, the efficiency is fairly low. So both have been looking at increasing the efficiency by adding factors to the initial GMT. And this is some of the work I did while I was a postdoctoral fellow in John Epstein's lab, where we reprogrammed tail-tip fibroblasts into induced cardiomyocytes using a 5-factor combination. And you see that these fibroblasts, after 2 weeks in culture, will beat. And in addition, this is a GCAMP molecule, which allows us to look at calcium cycling. And we see that in addition to beating, there's calcium cycling in these tail-tip fibroblasts. So again, I mentioned we were able to increase the efficiency of reprogramming, but it's still quite low in vitro. And again, it depends on if you look at expression of cardiac markers. For example, you'll see a higher efficiency, but I would subject that the field is going toward defining these induced cardiomyocytes based on more global electrophysiology changes, because these are functional measures of reprogramming. So most of the work that I'm I've been doing to date has been focused more on how do we make these cardiomyocytes become these specialized myocytes, either nodal cells or Purkinje cells. And I'm going to tell you a little bit about the work that first on neuregulin signaling. So while I was a graduate student in Glenn Fishman's lab, we discovered the CCS-Laxi line of mice, which labels the entire conduction system blue. So shown here is the neonatal mouse heart, where we can see the extensive His-Purkinje network in the ventricles. We can see the sinus node here within the right atrium, the AV node, the His bundle, and the bundle branches. And while as a graduate student, we looked at how the conduction system and when the conduction system is patterned during development. And it was thought that this happened very late in development, but when we looked at embryonic day 9.5, so here's a 9.5 day heart, where this is the common atria, this is the AV canal, the primitive left ventricle and right ventricle, we see the developing His-Purkinje system here between the left and right ventricles. And this is actually consistent with work that was done maybe 30 years prior to that by Virag and Chalice by electron microscopy, where they identified the developing His-Purkinje cells in this region. So this molecular marker recapitulated the developing conduction system. And when we look even one day later, we already see that these cells are beginning to be reorganized in the ventricles into this complex network that by birth resembles the His-Purkinje system. So this was a useful tool because until this point, it wasn't known whether mouse even had a His-Purkinje system because the heart beats so fast, and we were able to identify the His-Purkinje cells in larger species based on cellular morphology, but it's very difficult to distinguish these cells in the mouse heart by histology alone. So we use this mouse as a tool to study what factors can induce the ventricular myocytes to become the Purkinje cells during development. And one of the candidate factors that I was very interested in at the time is neuregulin, because neuregulin is in the right place and the right time to potentially induce myocytes to become conduction cells. So we know that the conduction system in the ventricles develops directly in contact with the endocardial cells, and we know that neuregulin signaling is in neuregulin, which is a secreted growth factor, was expressed by the endocardium, and the receptors, which are the ERB2 and ERB4 receptors, are expressed by the ventricular myocardium. And so we tested whether neuregulin could induce the cardiomyocytes to become conduction-like cells. So shown here is an embryonic day 9.5 heart, where we culture for 2 days either in the absence or the presence of neuregulin. And we see that in the presence of neuregulin, we see ectopic upregulation of the CCS-Laxi transgene here, for example, in the wall of the left ventricle and the right ventricle. So we've induced the expression of a conduction system specific marker in an ectopic site within the ventricle during development. And I should mention that this was only capable of inducing conduction cells during a very specific time point in development. So the plasticity for neuregulin to cause these myocytes to become conduction-like only existed during a specific time in development. So next, we wanted to see whether there was any functional changes in addition to the gene expression changes. And to do this, we did the whole embryo culture technique, where we isolate these early embryos and culture them ex vivo for 2 days, either in the absence or presence of neuregulin. And these embryos develop in they can develop normally outside the mother for 2 days. So you can add factors and test whether they influence cardiac development. And then we performed optical mapping studies in collaboration with Greg Morley's group to look at the results of neuregulin signaling in cardiac development. So shown here is a control heart, and we're looking here at a sagittal view where here is the AV canal, and this is the posterior wall of the ventricle, here's the apex and the anterior wall. And in control hearts, the cardiac impulse travels from the AV canal at embryonic day 9.5, along the posterior wall, then activates the apex, then the anterior wall. And this is consistent with where we see these precursors of the His-Purkinje system along the posterior wall in the embryonic day 9.5 heart. But when we culture these hearts in the presence of neuregulin, we see that the impulse travels not only from the AV canal along the posterior aspect of the heart, but in addition, it can travel along the anterior aspect of the heart as well, which suggests that we've changed the electrical phenotype of these anterior wall cells, and we've now recruited them to become conduction-like or His-Purkinje-like by the presence of neuregulin signaling. So Michael Flam's group went on to demonstrate in embryonic stem cells that the manipulation of neuregulin signaling can regulate cardiac subtype specification in human ES cells. So again, we can discover things in mouse, and they can translate into how to differentiate embryonic stem cells into these different types of myocytes. So when I began my postdoctoral training in John Eckstein's lab, I was very interested in other signals that may regulate the development and the programming of the conduction system. And the Notch signaling pathway was a candidate because actually Kellen McRae's lab demonstrated in fish that neuregulin as well as Notch signaling could regulate conduction in the fish. So I was interested in seeing whether it had an effect in mammals. And in addition, Notch signaling is involved in many important processes. It can take individual cells from within a population and specify them into particular lineages. And in addition, it can form boundaries between different compartments of the heart during development. So it's an evolutionarily conserved pathway, and we have, I promise not to go too much into the signaling, but I have to tell you a little bit. So there are 5 Notch ligands that are expressed on a signal sending cell and 4 receptors in mammals that can be expressed on the receiving cell. And when the ligand binds to the receptor, this intracellular portion gets cleaved and it goes into the nucleus. It binds to these mastermind-like factor and co-activators and activates target gene transcription. And so in order to test the role of Notch signaling in conduction development, we use 2 approaches, a gain-of-function model, where we can activate Notch signaling by expressing this active form of the receptor, the Notch intracellular domain. And I'll tell you a lot about activation of Notch signaling using that approach in the rest of the talk. And we can inactivate Notch signaling using a dominant negative form of this protein here called mastermind. So if we use a dominant negative form, it binds to the Notch intracellular domain, but it can't recruit these co-activators, and it inactivates Notch signaling downstream of all 4 of the Notch receptors. And then we use Cre-Lox technology. We clone this in such a way that we can express this activated Notch or inactivated Notch within myocytes in the heart. So for example, we can inactivate Notch signaling with this dominant negative peptide and mate these mice with Cre-expressing mice, which express within the ventricular and AV canal myocytes. And then we can study the effects of Notch inactivation in the mouse heart. So first, when we looked at the Notch inactivated mice, we found that they have a 50% reduction in the size of the AV node and a loss of connexin 30.2 expression. So connexins are isoforms of the gap junction channels within the heart, and we see that the expression of connexin 30.2 here in red in the Notch inactivated mice within the AV node was less than that in the control mice. So we saw a reduced expression of markers as well as a functional change on the AV node. So shown here is electrocardiographic EKG data from the Notch inactivated mice, where we see control mice have a PR interval of 43 milliseconds. The Notch inactivated mice have a significantly shorter PR interval, suggesting that they have defective AV nodal function. And when we looked at invasive electrophysiology, we looked at the AH interval, which is a more specific measure of AV nodal function. We saw significantly reduced AH interval in the Notch inactivated mice when compared with the control mice. And interestingly, when we looked at the AV node effective refractory period, we also saw a significantly shorter AV effective refractory period when in the Notch inactivated mice when compared with control. So not only are the nodes smaller, but they have a defective function, so suggesting that this may be regulating ion channels within the AV node. And we now know from genome-wide association studies that pathways that regulate the function of the AV node in the PR interval are often associated with arrhythmias as well. So next we looked at activation of Notch signaling in the heart, And shown here is a region of the control heart at the base of, here's the interatrial septum, here's the ventricle, and I've outlined the AV node here in white. This was demarcated by TBX3 positive staining, and we see here that the region of the AV node in the control mice is significantly smaller than in the Notch-activated mice. So when we inhibit Notch signaling, the node's small, and when we activate it, the node is large. So this led us to ask whether Notch activation can bias cardiomyocytes during development to become conduction-like. So can it take a non-conduction myocyte and turn it into a conduction myocyte? So one of the tools that we used to test this was the Contactin-2 GFP mice. So Glenn Fishman's group went on to use the CCS-LAC-Z mouse to identify a novel target of, a novel marker of the His-Purkinje cell, the Contactin-2. And this is a cell surface adhesion protein that's expressed specifically within the cells of the conduction system in the heart, but not in the ventricular myocytes. And so this Contactin-2 allows us to distinguish the His-Purkinje cells from the ventricular myocytes. So we looked in our Notch-activated mice to see whether we had ectopic Contactin-2 staining. So this would suggest that we've recruited the ventricular myocytes to become conduction cells based on lineage marker analysis. So shown here is a control heart. Here's the atria, here's the ventricle, and here's the Notch-activated heart. And we see Contactin-2 positive cells here in green. This is by immunohistochemistry. So we now have ectopic expression of Contactin-2, which is a marker of the conduction system. And shown here is a region of the interventricular septum where we see the right bundle branch, which is normally only subendocardial, only one cell layer. And in our Notch-activated mouse, we see ectopic expansion of the Contactin-2 positive cells. However, I will point out that these Contactin-2 positive cells were mostly expanded in regions near where Contactin-2 is expressed, so near the endogenous conduction system. And I'll get back to the reasons this may be the case a little bit later. So similarly, we looked at CCS-LAC-Z expression, which is another marker of the conduction system. Here's the right bundle in a control heart, and in the Notch-activated mouse, we see this ectopic expression and blurring of the boundary of the right bundle between the right bundle and ventricular myocytes. And when we section these hearts, we see that the right bundle cells in a control heart are normally only subendocardial, but with Notch activation, we can see these ectopic conduction markers more robustly. So now I've shown you that we have upregulation of gene expression that's consistent with a ventricular myocyte becoming a His-Purkinje myocyte. What about functional changes? So we looked at whether Notch is able to promote a Purkinje phenotype from ventricular myocytes. So first, this was published by Glenn Fishman's group. This is an action potential from a Contactin-2 positive cell, which is an adult ventricular myocyte adult Purkinje cell in a mouse. And this is a ventricular myocyte. And you can see that Purkinje action potentials are significantly longer, and they have this characteristic plateau in phase 2 of the action potential when compared with the working ventricular myocytes. So we isolated individual Notch-expressing cells, here where we labeled green with GFP, and we looked at the action potential of the Notch-expressing cells versus those of ventricular myocytes. And we saw that they had a characteristic action potential morphology that was similar to those of the Purkinje cells. However, the green GFP-expressing cells looked like the ventricular myocytes. And when we quantify this change in action potential duration, we see that with Notch, the action potential prolongs from 40 milliseconds to more than 90 milliseconds, whereas in the GFP control expressing cells, we saw no effect. So within this population, so Notch can prolong the action potential. Some of the cells were significantly prolonged all the way to that of endogenous Purkinje-like cells, whereas others were only partially reprogrammed. So there was some partial modification or extension of the action potential, but not a full Purkinje-like phenotype. So we next looked at the expression of the characteristic genes expressed in Purkinje cells when we activate Notch signaling. And this was activation of Notch signaling after birth So this would be reprogramming of those myocytes into Purkinje-like cells. We saw contactin 2 go up, which again is a specific marker of the His-Purkinje cells. We see upregulation of NKX2.5, which is a transcription factor that's known to be critically important in the His-Purkinje cells. We saw upregulation of HOP, which is also another transcription factor important in His-Purkinje cells. Of the cardiac connexins, which are the gap junction isoforms, we see upregulation of the His-Purkinje specific connexin 40 and downregulation of the ventricular myocyte connexin 43. We also saw upregulation of pacemaker channel isoforms, including HCN4, as well as the sodium channel subunit SCN5A. So up until then, these were the only known molecular markers of the Purkinje cell. So we wondered, well, was there something else that we're missing in order to make a fully bona fide Purkinje cell? So next we saw in some of the other studies that Ben Gillers has been doing that we see expression of canonical Wnt signaling within the developing His-Purkinje cells. So shown here in light blue is expression of axin 2 lax-E, which is a target of canonical Wnt signaling. And though it's faint, we did see that there is some expression of canonical Wnt signaling in the developing His-Purkinje system. And this is right at the time, embryonic day 14.5, where the decision between a myocyte and a His-Purkinje cell is being made. So we asked whether we could increase the efficiency of making these Purkinje cells during development by adding both Notch and Wnt signaling. So shown here is a control heart where we're looking at the left bundle branch. And here is a heart where we've activated both Notch and Wnt signaling during development. And we see ectopic expansion of the CCS-LAX-E transgene, suggesting again that these cells are reprogrammed based on lineage marker analysis to become cells similar to the conduction system. But in contrast to the Notch alone mice, when we just look at a region near the epicardial surface, very distant from the conduction system, Notch plus Wnt is able to upregulate this CCS-LAX-E transgene in this distant location, suggesting that it has an increased efficiency over Notch alone to produce conduction-like cells. And this work was done in collaboration with Stephen Springer and David Wang, who are in Gene Bones lab. So we next wanted to ask whether Purkinje-like action potentials, we next wanted to ask whether, in addition to the lineage markers, do we have functional effects that are additive with Notch and Notch plus Wnt? So when we looked at left ventricular myocytes, the control myocytes are shown here in black, Notch in gray, and the Notch plus Wnt in light gray. So we see a significant prolongation of the action potential duration with Notch plus Wnt when compared with Notch alone. So next, there are many things that can prolong the action potential. For example, we can either have increased sodium current or calcium current or decreased potassium current. So we wanted to ask what are some of the specific currents that may be regulated by Notch and Wnt signaling. So to do this, Stephen was isolating Purkinje cells, which are shown in white, and wild-type ventricular myocytes, which are in black. And he found that the peak potassium current in the Purkinje cells was much smaller than compared with those of ventricular myocytes here in black. And when we compared the Notch expressing cells and the Notch plus Wnt expressing cells, we see that they're becoming more similar to the endogenous Purkinje cells than Notch alone. Similarly, we saw he did not detect ITO fast in the Purkinje cells in contrast to those of ventricular myocytes. And the combination of Notch and then Notch plus Wnt is inducing the cells to become more Purkinje-like. We saw similar effects on IK slow, and as IK1, which was similar in ventricular myocytes versus Purkinje cells, was not regulated by Notch or Wnt signaling. So next, Aditi Chuplankar, who's a postdoc in my lab, wanted to ask what is the mechanism by which these electrical changes are occurring. And she looked at the expression of the transcripts that encode for the subunits that encode for the pore forming and the regulatory subunits of ITO, IKS, and IK1, and found that in fact, when we add Notch and Notch plus Wnt, we see a significant reduction in the expression of the subunits that encode for the ion channels. And this decrease in transcription was actually markedly paralleled the currents, decrease in currents. So it seems that at least in part, the decrease in current is modulated by transcription. And we are very excited about this because Notch signaling has also recently been implicated in a human disorder. HEY2 is the immediate downstream effector of Notch signaling. And it's been recently demonstrated that variants in the sodium channel locus as well as in HEY2 have been associated with Brugada syndrome, which carries a high risk of sudden cardiac death. And one of the things that we're interested in is whether Brugada syndrome can originate from this altered electrical programming during development secondary to Notch signaling. And so we're interested in understanding all of the ion channel subunits that may be regulated by Notch during development. So what I've told you till now is a story of cardiac development. And we know that the ventricular myocytes and the Purkinje cells have this plasticity that occurs until mid-gestation. But then after that, is there still a plasticity in the postnatal or the adult heart? For example, can postnatal ventricular myocytes become Purkinje cells? So the Cree-LOX technology I showed you up till now has been activating or inactivating Notch during development. So next, we wondered whether if we activate Notch in already differentiated myocytes, can we reprogram them to Purkinje-like cells? So to do this, we developed a system where we could activate Notch signaling in the postnatal heart and bypass its effects on cardiac morphology, but still allow us to analyze its effects on ion channel expression. And we developed this system using tetracycline regulated expression, where we can turn on Notch signaling after birth, but we can also turn it off. And this is important because when you want to do reprogramming, the definition of a reprogrammed cell is that it's epigenetically stable in the absence of ongoing gene expression. So this dual transgenic system will allow us to turn Notch signaling on, but also to turn it off and ask whether we've stably reprogrammed the cell. And so in this case, when we have the RTTA in dual mice that have the RTTA and the Notch1 intracellular domain, in the absence of doxycycline, there's no gene expression, but when we feed the mice doxycycline, we can turn on expression of Notch. So we can activate it and inactivate it in the postnatal heart. And so when we did this in postnatal mice, we saw that similarly, we topically upregulated contactin 2 expression and downregulated the same potassium channel subunits and regulatory subunits. And we've so far been doing this in the young mice, but we're now currently asking how late in development can we reprogram these myocytes. So we see both lineage markers as well as ion channel genes changing. And why is this significant? In addition to regenerative medicine approaches, in injury responses, for example, in the liver, when hepatocytes are exposed to viruses, they activate Notch signaling. And these hepatocytes, when Notch is activated, form biliary cells. And these are the same signaling pathways that regulate hepatocyte formation during development. But when Notch signaling is turned off, these hepatic progenitors make more hepatocytes. And it's been shown that the blipping on of Notch in this response in the liver to viral pathogens can cause cell fate changes and reprogramming in injury responses. And this is interesting because Notch signaling is actually activated within the ventricular myocytes in myocardial infarction. And it's thought that this activation of Notch signaling does a lot of good things in the heart. For example, it can promote cardiomyocyte progenitor cell proliferation and neovascularization and prevent cardiomyocyte apoptosis. These are all good things if you're having a myocardial infarction. But it may also be that its regulation of ion channels in ventricular myocytes may cause arrhythmogenesis. So, for example, if Notch signaling can regulate potassium channels as well as cardiac connexins in the adult heart, in addition to promoting all those good things, it may provide a substrate for arrhythmias. So some of the current and future directions for this part of the project, we're looking currently at whether the Notch-activated ventricular myocytes, and we're using a special mouse that enables us to specifically look at which cells activate Notch signaling after myocardial infarction, whether these are reprogrammed and whether they downregulate connexins or regulate ion channel expression. We're also currently looking at whether the expression of other ion channel subunits, for example, calcium and sodium, are regulated by Notch and Wnt signaling in the ventricular myocardium. And we're interested in whether this activation of Notch signaling in the ventricles has functional consequences at the whole organism level, for example, an increased susceptibility to arrhythmias. And also, Aditi Chiplankar is looking at the transcriptional and epigenetic modifications that occur downstream of Notch and Wnt signaling. So I've told you about what happens when we activate Notch in the ventricles. We reprogram these cells during development as well as postnatally into Purkinje-like cells. But what about in the atria or the sinus and AV node? What happens when we activate Notch signaling in these areas of the heart? So there was a recent paper that just came out that showed that Notch signaling, when it's expressed in, so as I told you before, these sinus node cells develop from the posterior second heart-filled progenitors. So here's a primitive sinoatrial nodal cell. And in the presence of Notch signaling, it's been shown that during development, this transitions the sinoatrial nodal cells into atrial working myocardial cells. And Notch is normally kept from preventing these sinus node cells from becoming the atrial working myocardium by the expression of MKX2.5 within those cells. So we next asked what happens in the atria when we activate Notch signaling after birth. So these mice develop normally. These are the double transgenic mice. We can keep them in the absence of Notch signaling by providing no doxycycline, and we can turn on Notch signaling by giving them doxycycline in the chow. And so we activate Notch signaling here at several weeks of age, and when compared with the control mice that have normal pairing EKG with P-wave and QRS, when we activate Notch signaling in the atria, we see a sinus bradycardia, so slowing of the interval between the P-waves, as well as AV block. So here's a P-wave without a subsequent QRS complex. So we see sinus bradycardia and high-grade AV block. So we took this cohort of mice that was not on doxycycline, so no Notch, and then we gave them activated Notch for 7 days, and we saw the heart rate went from 569 to 328, and the PR interval significantly prolonged from 36 to 53 milliseconds. And in addition, a small subset of them went on to have AV block after 1 week. And when we looked at the morphology of the sinus node and the AV node, based on TBX3 expression, we see that the cells are still present in the Notch-activated mice. So here is the TBX3 positive AV nodal cells. So it's not that the heart has just disintegrated or that the AV node is structurally abnormal. This is a functional electrical remodeling of these cells. So as I mentioned before, if you reprogram something, when you turn the gene expression off, you should still have a stable phenotypic change. So this is in the absence of doxycycline, and then the presence of doxycycline. I just told you we have a sinus bradycardia and AV block. And then we continued half of the cohort on doxycycline, so continued Notch expression. And in the other cohort, we removed doxycycline. And we saw that in the presence of activated Notch signaling, the heart rate, in fact, we went on to have atrial pause. So we had a complete silencing of the sinus node. And interestingly, even in those mice where we removed doxycycline and turned Notch signaling off, we still see that the mice went on to develop a complete atrial pause. So this is the definition of reprogramming. We've reprogrammed the sinus node cells. And this is the work that's been done by John Chow, who's a student in my lab as well as Igor Efimov's lab, and in collaboration with Bas Balkans, who have looked at, the question we had is, well, we don't see a P wave, but we can have an absence of a P wave if the sinus node doesn't fire, or if it doesn't couple to the atrial myocardial tissue. So which is it? So here are the control hearts, where we see atrial to every ventricular impulse. And in the Notch-activated mice, here we see one P wave, and then we see a QRS complex. And when John looked at the activation maps of the atria, he found that there were no instances where we saw firing of the sinus node that it didn't couple with the atrial tissue. So it seems, in fact, that the sinoatrial nodal cells and the AV nodal cells are being reprogrammed and are no longer having pacemaking activity. And he was able to partially rescue the sinus bradycardia by giving isoproterenol, but not fully rescue. So here are control hearts in sinus rhythm and after isoproterenol, and then these are the Notch-activated hearts in the atria and ventricle, and either before or after isoproterenol. So the longer we turn on Notch, we continually remodel the atria, not only the sinus node, but the atrial myocardium as well. And that's represented here by a prolongation of the conduction velocity. Here we see very fast conduction within the control atrial tissue, whereas when we activate Notch signaling within the atria, we see that it takes significantly longer for the atria to activate. And we're currently looking at the gene expression changes, and we have some ideas of what's going on downstream of Notch activation in the atria, but that data is not yet ready for prime time. So. And in the last 3 or 4 slides, I want to tell you how we may leverage these transcription factor networks for regenerative medicine approaches. For example, to produce a biologic pacemaker. So what is a biologic pacemaker? So a pacemaker would be a tissue that can spontaneously or via engineering confer pacemaker properties to regions of the heart. And several groups have tried to generate biologic pacemakers using cell-based therapy. For example, we talked about before how it's possible to manipulate pluripotent stem cells ex vivo to become pacemaker-like by manipulating neuregulin signaling. And then you can inject these cells back into the heart and hope they couple and produce a functional pacemaker. And there has been some, as you know, those who were at Michael Flam's talk can attest to, there's been some progress made on this type of approach. Another possible way to produce a biologic pacemaker is to do gene therapy using, for example, adenoviral vectors to deliver specific channels to region of the heart that can confer pacemaker properties to the heart. However, this wouldn't produce a complete phenotypic switch, and you'd maybe get 1 or 2 of the ion channels important in the sinus node, but not all of the ion channels. So the method that I think is most promising is directly converting the myocytes into pacemaker cells by expression of transcription factors. And along these lines, it was recently demonstrated that TBX18, which is one single transcription factor, can take ventricular myocytes and reprogram them to sinoatrial nodal myocytes. And this was done in mouse as well as guinea pig hearts. So shown here are ventricular myocytes expressing a controlled GFP, green fluorescent protein, TBX18 expressing ventricular myocytes, which have now adopted the spindle-shaped morphology characteristic of endogenous sinus node cells. And they went on, this was recently published, not by us, but I thought it was very exciting that they showed that TBX18 can convert these myocytes to make pacemaker cells not only by morphology, but in addition, we can now see that they have spontaneous action potentials similar to those of sinoatrial nodal myocytes. So one of the things that we'd love to try is to do this in human tissue with TBX18 as well as possibly other transcription factor combinations. And one of the ways we can try this is in organotypic cardiac slices, and because in contrast to those of single cells, the slices can provide a tissue-level electrophysiology that we can't get on single cells, and as well as they may provide an ability to have long-term culture, because it may take maybe days or even weeks to take a ventricular myocyte and make it be reprogrammed to a conduction cell. So in mouse, this takes about 2 days to be from a ventricular myocyte to a conduction cell. And in humans, these processes typically take at least twice as long. So we need to be able to culture these cells at least probably for 4 days to be able to assess these types of changes. And so this is data that was generated in Igor Efimov's lab by Chao Yi, John, and Patricia, where they took these slices of ventricular myocardium and analyze them with multi-electrode arrays. Here we see across the whole slice, as well as single electrodes. And then they see that these slices have typical anisotropic conduction of ventricular myocardium. So this is really exciting and may provide us with an opportunity to use viral-mediated reprogramming in humans. So with that, I'd like to acknowledge everybody, because obviously I did not do all of this work alone, and I've had the fortunate ability to work with some very talented and wonderful mentors. First, I'd like to acknowledge my laboratory, Aditi, who's a postdoc who's working on the Purkinje cell reprogramming together with Stephen and David in Jean Nerbone's laboratory. Ben Gillers, who is working on the ventricular pre-excitation project, and I didn't get a chance to tell you about that today, but he's found some very interesting findings that Notch is regulating Wnt signaling, and that's involved in the downstream signaling for pre-excitation phenotypes. So hopefully he'll have a chance to present his findings at Research Day, and you can look for that. John, who is a student who's in my lab as well as Igor's lab. Stephanie, who's a technician in the lab and who is involved in all the projects, and Kate, who will be joining the lab soon. Igor's lab, I mentioned all of the people who collaborated in Jean's lab on the Purkinje reprogramming project. The viruses are being produced by David Curiel and Igor Dmitriev here. I didn't tell you about some work we did in collaboration with Don Elbert's lab, and I'd like to also acknowledge some of the published work where we collaborated with John Epstein, Vic Patel, Glenn Fishman, and Greg Morley, and I'd like to acknowledge my funding sources. And I'll take any questions.
In the early slides where you showed the staining of the Purkinje system, I noticed the whole right atrium was dark. Is that artifact, or is that actually the whole atrium is stained?
Yeah, so that CCS laxi mouse within the atria is not specific for the conduction system after birth. So in the ventricles, it's only expressed in the conduction cells, but in the atria, it's expressed at high levels in the sinus node and AV node, but it's not specific. So there is some expression in the atrial myocardium. So if one wanted to specifically mark the sinus node cells or AV node cells, there are other markers now that are much more specific to those cell types.
I have a somewhat, I guess, off-topic question. So with regard to reprogramming the cells in vivo to like ventricular myocytes or fibroblasts to a conduction cell phenotype or to a ventricular myocyte phenotype, I was wondering if you had any thoughts or if there's anything known on what taking that cell from a fibroblast to re-induce it to a different phenotype, taking that through that stage, if there would be immune response to that, that wouldn't be typical because that wouldn't happen in a normal adult.
Okay, so the question is whether if you reprogram in an adult, whether that would cause an immune response?
Yeah.
Well, I think some of the immune response it depends how you reprogram. If you're using a virus, then the virus itself could cause an immune response. But since those would all be endogenous genes that are turning on, I don't suspect that per se would be immunogenic. But if you use an adenovirus, for example, so these are mice, and we can do make transgenic mouse models and manipulate gene expression without having to actually do gene therapy approaches. But if you were to inject a virus, like an adenovirus, most humans have been exposed to adenovirus, so they would mount an immune response. Now, David Curiel and Igor Dmitriev have, as well as others, some ways to get around the immune response. There are different ways to make the adenovirus that it would evade the immune response so that you may be able to actually do gene therapy. And this has been done using these modified adenoviruses in cancers, for example, to treat, to give gene therapy to treat tumors. And those are in clinical trials. So it's possible.
But would the cells themselves express anything that they wouldn't normally in an adult cell that would be immunogenic?
I don't think so. Not that I could think of. I mean, there are Purkinje cells, in the adult, so, in the heart. So I don't know that upregulating any of those genes would necessarily produce an immune response.
Yeah, the one slide had 2 time scales. It looked like there was 1 pulse every 3 pulses on the other one.
Which one was that?
It was the one where you had, it looked like graph paper with pulses on it, and one of them was...
Oh, like a telemetry strip? Mm-hmm.
There were 2 of them in comparison. It looked like there was a 3-to-1 difference in the time scale. Yeah, that one. So it looked like there's a 3-to-1 difference there. Are those the same time scale or different time scales?
I think they're the same time scale, yeah. Mm-hmm. Yep.
There's 3 difference in the time scales, okay.
Yep. Mm-hmm.
So I'm not a mouse physiologist. So when a mouse is born, it lives, what, about a year or 2? Do mice live that long?
It depends on who's taking care of them. Probably at least 2 years.
So if they don't meet a cat, they live.
That's right.
Okay. So after the mouse is born and it has its conduction system, does the conduction system, like other nerves in like humans, when the claim is that your nervous system doesn't replace itself. And so does the same thing happen in a mouse conduction system, that if you do no experiments in the mouse, just normal physiology, is there a turnover of the Purkinje cells in a mouse? Is that known?
I don't know that anybody's directly looked at that, but I would suspect that it would mirror that of what happens to other myocytes. So in the absence of injury, you probably don't have much turnover of those cells. But to my knowledge, nobody has directly looked at that. So you could potentially label all of the cells at birth and then see...
And then see if it's the
later.
same stuff 2 years later.
Right. I mean, that experiment could be done, but I don't know that anybody has done that.
So you see why I'm asking. So I'll tell you why I'm asking, because I don't know anything. But the way I was thinking of it, if there's actually a natural turnover, say slow, eventually maybe every 6 months or something, the things replace themselves, then it's conceivable that when you're inducing things, myocytes turning Purkinjes, is it not possible that you're waking up the sleeping cells that were intended to replace the turnover cells?
Right. I mean, this is definitely a possibility. If you were to think of it that way, one would say, how did they become, subendocrine? You mean specifically in the studies that we're doing, or?
Well, I'm asking just basic physiology. If there's a turnover of cells, which happens, as far as I know, in most living things, then is there a way to check to see that when you activate things, that you're not essentially waking up the cells that were already programmed or predestined to be the replacement cells for the cells which are alive today? That's all I'm asking. I don't know if that's possible or not.
So you could do lineage tracing studies. I mean, it's been done in humans as well as mouse. For example, myocytes do turnover, but at a very slow rate. There's very slow regeneration in normal human and probably mouse tissue. There's probably a way to boost that, and there are a lot of people working on, how to make a myocyte proliferate after, in an adult heart. But we are not doing that per se when we activate the signal.
So the basic question, so it is known that the mouse conduction system that a mouse is born with, in principle, if you labeled it and put an X on it with a dye or some molecule or something, then when the mouse died 2 years later, it's the same cell.
I don't know that anyone's done exactly that experiment, but the conduction cells actually proliferate very slowly, and so
But there's no turnover.
So it's really thought that the cells that you're born with are what you die with, but plus or minus 0.0001.
Yeah, no.
I mean, so.
It doesn't matter.
Okay, so I'm a little confused there. So please just enlighten me. So if you have, convert some of your cells into conducting cells, wouldn't that actually create certain for arrhythmia?
Arrhythmias?
Yeah. So this is something.
So these mice do get so I didn't tell you the story about the atrial arrhythmias that they get.
Yeah.
And so it's true, you would think that they're arrhythmogenic, and they may be. You may just need to stress them in order to bring out the arrhythmogenicity. So, mice are not the greatest model of arrhythmias. They don't, you may need to kind of program stimulate them or, to induce an arrhythmia in the setting.
Yeah, so your purpose is to repair the conduction system, right?
Right.
But I mean, the repair. So you all of a sudden you change. You introduce some changes that, to me, it is just just putting in the substrate for arrhythmia.
Mm-hmm.
And what is different than what people use? They use the biological
But
pacemaker.
let me ask you, the purpose would be to regenerate the tissue, but also to understand normal disease processes. So for example, we know in myocardial infarction that these signaling pathways go up in the myocyte, and we know that that produces arrhythmias. So it's possible, in the understanding of development, that we can understand adult disease and maybe harness that for regenerative medicine in a very controlled way. I think we're still quite far from the regenerative aspect of it, but you're right in that if we just start expressing these transcription factors, in a large animal, we may produce arrhythmias and no Purkinje cell.
Right. So now, actually, I'm confused because I always accept that biological pacemaker will be it's a nice thing. But then would that also generate the arrhythmia as well?
I mean, so these studies, the TVX18 paper is very recent, and it hasn't yet been replicated. So we don't really know what will happen. I mean, I think that the promise is that myocytes have a plasticity that we didn't recognize from, to be one. We thought that if you were an atrial myocyte, you're an atrial myocyte, and there's no way that you can become a nodal cell. Or if you're a ventricular myocyte, you're a ventricular myocyte, and there's no way you could become a nodal cell. But we now understand that utilizing, these developmental pathways, that there may be a plasticity in the adult heart that we didn't appreciate before. This may be important to understand disease or regeneration, but I don't know. I would love to do some of those studies, but.
But I think what if I can translate, I think the idea is if you have ectopic loci that are themselves pacemakers, wouldn't you predict that they would be pro-arrhythmic? And I think the answer is yes. I mean, people have done that. But I think what you're leading to is the idea that you'll be able to do localized focal reprogramming in a particular region of the atrium.
Exactly.
And generate a new SA node.
What
or
happens
AV
to
node.
the original SA node?
Well, there are
So
some.
you would ablate it, just like you do in a person when you put a pacemaker in.
So that you have to have the perfect timing. That when you put in, you have
No
to.
question about
Right.
that, but I think that's the approach, that's the only way you will get away from the pro-arrhythmic idea of just reprogramming at random. That's the only way, is to be able to do it in a highly localized, specific way.
There's this sick sinus syndrome that occurs where in the adult, there is an absence of pacemaker cells. They're replaced by, fibrous tissue. So if one, in the setting of this type of patient, if one could produce a pacemaker cell instead of putting in, an electronic pacemaker, so it would have to be, as you said, very focal. So you'd have to have delivery methods to make a very focal pacemaker.
I thought the electronic pacemaker is just, it can take over. It's just, with the biological pacemaker, it has to grow, and then during the, so
But
it's
actually,
not-
it doesn't
yeah.
have to grow, right? Because if you take a cell that's there, okay, and you turn it into something new, it doesn't have to grow, it just...
Yeah, but you have to make sure that all the cells are turned into that, so that at the same time. So biology is not that homogeneous. So
Right,
that's the
right.
problem, right?
So this isn't going to happen in tomorrow, but hopefully,
Yeah.
in my lifetime, I'm still fairly young.
But, I just want to know if it is a problem, right? Because I always accept
You're right.
it, yeah. All right.
There is definitely a long way to go.
All right. Thank you.
Yeah, this was a great talk. I have one question. I just want to know your thoughts about the lifespan and the quality of these transformed myocytes.
So the lifespan of the
Yeah,
myocytes?
and the quality of these, the transcription factors in these transformed.
In other words, are they happy?
Yeah.
The quality of their life?
Yeah.
I don't know.
It's going to be certainly tough.
I don't know that I know whether or not they're happy. I can only tell you what I already know. But what we do know is the
Action
lifespan.
potential, you must have looked into the action potentials, right?
Mhm.
So are these action potentials comparative to the original Purkinje part of the cells action potentials in terms of their quality? That's what my question is.
So some of them look just like a Purkinje cell, and we would call those fully reprogrammed, right?
Yeah,
So,
that's what it is.
and some of them aren't quite, and some of them are more similar to a ventricular myocyte. So
Right.
once we understand the signaling pathways that make these kind of cells, then we can understand why some of them are not fully
Right.
Purkinje cells. So I think we may still be missing, some factor where not 100% all Purkinje-like. So, it may be that there is an inhibitory signal that's present in some parts of the heart saying no Purkinje cell can form here, or it may be that there's another cooperative signal that is cooperating together with what we already know. And I think, there are multiple ways that people do reprogramming. Some is just try everything, screen 100 factors and every combination, and then there are kind of hypothesis-driven. And I've sort of taken more the hypothesis-driven approach where we see, okay, we activate this factor, we look at all the factors downstream of it, and what have we missed? What is missing in that cell type? And if we can identify something that we miss, add that, and see if those factors together are better. And then if they don't make a full cell, why not? What currents do we not have? And what regulates those currents? So we don't yet have all the answers, but I think it's really exciting that, we can start to see that we can change these phenotypes even in the adult heart. And so I think it's still at the very infancy of, of being able to do these studies.
One final question about external work. Myocytes, if you look at them on a microscope, you can put a little load on them and they'll move and wiggle and move things around. And so what happens to the machinery within the myocyte once you reprogram it? Is it still contractile? Does it still do external work?
So the Purkinje cells do contract, but they have less force generation. We haven't really looked, they downregulate, they have downregulation of sarcomeric proteins and, differences in T2-butyls and glycogen content. What I've shown you is all of the ways that we've assessed them, but we haven't looked at the contractility, but that's something that we could definitely think about doing. Great, thank you.
Thank you.